Hello, my name is Dr. Varun and you are watching Chlorophyll classes on YouTube. Well, in this video, this is for the 12th class students and we're gonna be studying about the principle of inheritance and genetics. The topic that we're gonna be doing is the linkage and the recombination. Well, this is one of the topic which most of the students from board level or maybe as the NEET level, they afraid and they always pray that this never comes in their examination. But here at the Chlorophyll classes with me, we will be debunking all the issues and try to make it as much as interesting, knowledgeable and understood to you. Let's just start with it. Well, linkage. First of all, you have to remember that who studied about it. This thing was studied about T.H. Morgan. Okay, so this T.H. Morgan studied about it. So it is studied by T.H. Morgan. Okay, this thing, let's just deeply go slowly into the depth. Now, what happened in this? Linkage word itself says that something is linked with something. Okay, for example, if I am there and with me over here my friend is there. So wherever I will go, my friend will also go with me. So this happens in the classroom. Now, like one student goes, sir, I am going to washroom. The friend, the bench partner is also like, I am also going, sir. So this is what linkage. Same thing happens with the genes. What happened? When the genes which are closely present, I call them the genes which are closely friends, they will become close friends. So the close friends, when they have to do something, they will do together or they will go together. When they will go, okay, the major question is there. So let's just go back to the meiosis from the 11th class. In meiosis, what happened, beta? We did something like that. This is what? Chromosome, like homologous chromosome. What happened on this chromosome? I told you, na, there will be what? There will be, these are what? These are its, okay, let's just increase it. So these are what? These are the different genes. Yellow color genes, this color gene. Now over here, this yellow color gene is little different. Here it is this like this. And the green color one is also little different shade because different allele is there. Of course, the gene will remain same, just the allele difference I am showing it with the lighter or darker color. Okay, capital small, remember it like that. Okay, now what happened, beta? The thing is that when these two things will cross over, right? So what will happen? When the crossover will happen, this thing will go to the this one, this thing will go with the this one. Now you will say that, sir, why two genes are going? Because they are closely ringed together. Means they are what? One friend, one friend, one friend, one friend. So they are close friend, close friend. They go to this side. This toilet is this side, this toilet is this side. So they went like this together. Okay, so friends are going together. This is linkage showing. I hope something is understood to you. Okay, let's just come back to our topic, okay, and understand it much better. Okay, so what we will remember that first we will see the linkage, what it is. So linkage is the tendency of genes, because I told you na, genes will go together. Okay, genes to stay together during inheritance throughout generation without any crossing over. Okay, because I told you na that colors can cross over. Okay, but here these genes do not cross over, means new recombinants are not being formed in their cases. Slowly you will understand. Okay, don't worry. Without any change or separation, due to their being present on the same chromosome. or chromatid. Okay, better word is chromatid. Okay, now we have understood something. Let's just go into it. Now, what would be it that, okay, we understood that this thing is there. So which genes would be linked and which would be the difference between the genes? So broadly, we can change the genes, like we can classify the genes into two types: linked genes or unlinked genes. Linked genes which are closely present, unlinked genes which are not. Like for example, my friend is going with me because he's closely linked with it. But what if the friend who is there, who I don't meet, who I don't see, so that person is not my friend, so I won't see, I won't like do the linkage kind of thing. So with me and him, no linkage is there, or very less linkage is there. Okay, so this thing is what, on this basis we can separate the genes into two types. Here we will understand something and later we will see the lot of points related to it. So they are what, we have linked genes and we have unlinked genes. Unlinked genes. Now the linked genes are what, which are closely present together. Genes located on same chromosome. And unlinked means which are related on the different chromosomes. Okay, so they are not that close. Okay, so these are what, genes present on different chromosomes. Now when these linked genes are there, okay, normally what happen in unlinked genes? Unlinked genes means whatever happened in the Mendel heritage. So what happened in the Mendel inheritance? In Mendel inheritance, we see that two genes, when they are coming up, they are showing the new forms: parental combination, recombination, non-parental combination. We studied now, same thing. So what happened in the unlinked genes, when they will undergo selfing and those things, there will be new transformations that will form. So we will see that, write it up. So what will happen? It shows dihybrid cross, as when there is a dihybrid cross will be done 9:3:3:3:1, and if we do the test cross, everything will come 1:1:1. Okay, and dihybrid test cross as 1:1:1:1. Here in this cases, 50% would be parental, 50% would be non-parental or the recombinants. Okay, so what happened? 50% parental and 50% recombinant. This is only we used to say that. Okay, but what happened in case of the linked genes? Here they do not, when there is a dihybrid cross in the linked genes, means the genes are different. So two genes are there, means two genes inheritance, means dihybrid cross. When we do the dihybrid cross, they will act like a monohybrid result, means 3:1 result. So what happened? So dihybrid cross shows as 3:1. That is like monohybrid cross result, like monohybrid cross. We will see that. Okay, here we have to just remember the fact, monohybrid ratio, and the test cross would be what, 1:1, and test cross will be 1:1, means both will be the parental combinations, no recombination forms. Okay, so all 100% parental combinations, no recombinant scenes, no recombinant scene. Okay, we will understand it with the graph later on. For now, let's just see it. Okay, we understand this thing and this basic something. But this thing must has been worked by the T.H. Morgan. How he do it? So he did it with the help of a organism, and this time it is a animal. And which animal he has taken? He has taken a from arthropoda fruit fly, which is known as Drosophila melanogaster. This Drosophila melanogaster is also called as jackpot of genetics, because what all the features it show is similar like what the features has been seen in the pea plants. So we have taken the pea plant for certain reasons. Same way we have taken this also for certain reasons. So we will learn that. Let's just see it. Okay. So Morgan took fruit fly, which is called as Drosophila melanogaster, as his experimental subject, for genetic material or for genetic studies. And the reason why he did it, we will later on, and that's why it is called as what? Jackpot of genetics. Okay, now let's just see that why he did it. So question is this, answer is that, first of all, this fruit fly has a very small lifespan of 2 weeks. So within 2 weeks it grows, reproduced and die also. Okay, shorter lifespan means, in shorter lifespan means lots of progeny can be seen. So means if I start the experiment today, within the 1 week I am able to get lot of progeny from it, and then the next progeny also, like babies will grow by that time and I can see them. So multiple generation I can easily analyze. We can't analyze our generations because one life is 80 to 90 years old, okay, in our country as of now. So what happened that we can, maximum person can see the grandparents same or the children's. That's the maximum I can study when the human I am taking as a subject. But this has only 2 weeks, so I can see multiple generations and I can see the inheritance pattern. Understood? First is short lifespan of 2 weeks. Second, easily cultivable. Easily cultivable means what? That you can easily, like, it feeds on what? A basic medium. You don't need any fancy food or anything, so easily can be fed. Then it produces lot of progenies in one mating, means hundreds of progeny are being formed, eggs are being released and they will be formed. So lot of progeny means last probabilities, all the possible probabilities are being shown. Of course all the possibilities can't be seen, but maximum of probabilities, all the possible probabilities can be seen into it. Okay, and it is easily available. Fruit fly is available on the fruits, okay, because they are like fruit, we want fructose kind of a thing, sucrose. So they are available easily, so not a very fancy thing we have to find and tough to find. Okay, so what are they? They are easily available, then easy to breed or cultivate, short lifespan of 2 weeks, so easily can be seen that the multiple generation, so multiple generations can be analyzed. Then single mating produce multiple offsprings. Single mating produce hundreds of offsprings. And then one more thing is that male and female is easily distinguishable, means sexual dimorphism is there. So it help in what? It help in finding, okay, male characters, female characters, something is related to very much male, something related to very much female, means linkage is very easily established over here. Okay, and inheritance patterns can be seen in males and the females. Okay, so that is what males and females. Females are distinguishable than males, because males are smaller, female are large and bigger and broader in size. I hope this thing is understood to you. Let's just see that what all the contribution Morgan did. He did experiment, okay. What are the contribution he did? He did it for the linkage and all those things, but with it what it is did. Okay, so what happened? This is the important thing, contribution of Morgan. First of all, he is called as father of experimental genetics because he did the experiments. Also the fly man of the genetics. Okay, so it is called as, he's the father of experimental. We will see that what he did. Okay, don't worry. Whatever the crosses it makes, same like the Mendel, he did crosses. We will see. Experimental genetics, or called as fly man of genetics, because fruit fly na, so he cultivated it, so fly man. Okay, so fly man of the genetics. Same like if you have seen the Robot 2.0 movie, here also what that, the Birdman, same fly man. Okay, so he discovered about the crossing over and the linkage. Okay, he discovered linkage and term coined for also it. Termed coined also. And he even see that how that linkage is not taking place in unlinked genes. Okay, lot of things are there, we will learn it. Okay, so that thing he found it by what? He saw the chiasma. That chiasma is something related to the recombinations. So if you remember the meiosis, go back in the prophase 1 and remember the facts about it. If not, then you can even go back and see the 30 days 30 most important topic where I was talking about the prophase 1. Okay, you will understand it. Okay, so for now, okay, for 11th class we will do it when the cell division chapter will come. Okay, so third thing is showed chiasma causes crossing over and the recombinant comes. Okay, since there are certain cases where the chiasma forms but the recombination is negative because close linked genes are there. Okay, next thing is gene mapping, and on that basis the genes which are closer present, he was like, okay, this is close, this is close, this is close, or this is not close. He are able to make the map of all the genes which are present on the chromosomes. So right now, like when I say that the chromosome, this has this gene, then this gene, then this gene, who made that? He only found out, and we have to learn it. It is a very important conceptual numerical based questions to it. Okay, let's just see that what is the next thing, that is a linkage groups. Linkage groups means that how many chromosomes are present, that represents the linkage groups. Means on one chromosomes there will be multiple genes present into it. It represents one linkage group which has so many genes which are linked together. Okay, so number of pairs present, number of linkage groups would be present. Means in my body 23 pairs of chromosomes means 23 linkage groups present. In P7 chromosomes present means 7 linkage groups present. Means one chromosomes has how many genes present? They are all what? One linkage group. Clear? Okay. So next thing is linkage group. Linkage group. So in linkage group, it is what? Genes that are present on the same chromosome makes one linkage group. So this will be direct question to you that in humans, in fruit fly, how many linkage groups are there? So you will remember that fruit fly has 4 pair of chromosomes only. So 4 linkage group. So fruit fly, 4 linkage groups. 4 linkage group as it has 4 pairs of chromosome. So one homologous chromosome considered as one linkage group. Okay, so it can also be represented as 4 homologous chromosomes. Okay, next thing, in case of humans, how many pairs we have? 23. So there will be what? 23 linkage groups. Human has 23 linkage groups. Then we have P. It has 7 linkage group. Now, linkage group size matters. Okay, so if a chromosome is longer in size, means has more genes, means big linkage group. Smaller will have smaller linkage groups. So next thing which I am writing is that size of linkage group depends on the size of chromosomes. Like Y chromosome has 231 genes. Chromosome 1 has 2969 genes. So this is what? It is a large linkage group. Multiple genes are there. And this is what? Small linkage group. Because less genes are present on the chromosomes. So small linkage group. And what is the problem with the linkage? Now think that me and my friend, okay, he loves wherever he will go, I will go, wherever I go, he will go. So we are linked. So means, for example, I am good in biology, my friend is good in chemistry. But when the class is being asked, so we both, like it's a biology class, we both are going, chalo, okay, let's go, po, okay, so vange, okay, so everyone is going. So the teachers would be like, what? Hey, why you are coming, chemistry student? You are not even good enough in biology. And same goes with vice versa. And this becomes what? Headache. The teachers are what? Imagine the teachers are the farmers who will cultivate the crops. They want my gene to be participating, but this is coming extra problem which we don't want. So breeders, the cultivators, the farmers has a major problem during this time because non-desirable my friend is also being the part of it, of the crop. Okay, so in the crops we want only certain features, we don't want other features, but it is coming up because of the linkage. Okay, so we will remember this fact that linkage is a biggest headache for breeder because it does not allow them to freely bring all the desirable characters, or we call it traits, in one variety. Okay, now after this the next thing would be the arrangement of linked genes. Okay, now sometimes what happen, like the chromosomes are there. Okay, so sometimes what happen, this is the what? Homologous chromosomes. Maybe this chromosome has both the dominant allele, and this is both the recessive allele. So this is one type. Another type is what? That this has one gene dominant, other gene allele recessive. This is recessive, this is dominant, like this. Means vice versa is there. But since they are what? Linked genes. So there are two different arrangements are there. And if you remember organic chemistry here, you will remember something would be cis, something would be trans. So cis would be what? Same side. Cis what? Same. Okay. Trans means opposite. Okay. So we will learn it. So what it is? Arrangement of linked genes. Arrangement of linked genes. In arrangement of linked genes, there could be of two types, cis arrangement or trans arrangement. So we have what? We have cis arrangement and we have trans arrangement. So imagine that this is a chromosome. Now we are coming to back the genes level and the alleles level. So imagine that this is the gene A small and here also capital small. So what is happening? In this case, dominant allele, both dominant alleles, like we are always taking with respect to two genes only, okay? So both dominant allele are present on one chromosome and recessive allele on homologous chromosomes. Homologous means similar but other. Okay. Homologous chromosome. Okay. Now in this is the first case. Second case is what? A is there like this, but B goes like this. Capital is on the second one and here we have this. Okay. So in this case, what happened? This called as trans, not the same. So the same and this is being taken with respect to what? It is taken with the dominant allele or the recessive allele. So if the dominant and recessive allele are on the two different ones, separation, then it's okay. But if one gene's dominant other genes on other, it will be what? Trans arrangement. I hope you would have understood it. Okay. So here what? One dominant allele is on one chromosome and other gene dominant allele on other. Vice versa for recessive alleles. This is cis and trans. Next comes the type of linkage. So linkage is two types, complete linkage, incomplete linkage. Let's just study about it and see the linkage in more depth. Okay, so next thing is types of linkage. So we have what? We have two types, that is complete linkage and another option is incomplete linkage. Now here you have to understand certain fact beta. What is the fact? That complete linkage is very rare. Okay, we don't actually find complete linkage. There is always incomplete linkage. Means, let's just go back to this point. Okay, if you remember this thing, what we did? We said that the genes would be showing this two different effects: unlinked genes, linked genes. If it will be complete linked, this behavior would be shown. Means even if there are dihybrid, means two different genes, but the inheritance pattern would be like one, that they are a single unit. Me and my friend is single unit, combined effect from with shampoo conditioner free, like this. Okay, but unlinked, like incomplete ones will not be completely like this, will not be completely like this, somewhere in between this, but effect would be closer to this. Means there will be a recombinations there, but in between this, that is an incomplete linkage. Did you understand certain facts? Let's just see the complete linkage and incomplete linkage in detail. So starting with a complete linkage. Okay, so first of all, the whole progeny, shows parental combination. This we already know because no recombinants would be there. Okay, so whatever we know, we are writing it. Parental combinations. Then complete linkage is rare in the nature. Complete linkage is rare in nature. Now, if someone will ask that why it is rare in the nature, because linkage leads to less survivalhood. Why? Because variations won't come up. If the variations are not come up, then any effect would lead to two important features going away from us. Okay, we learned it in the evolution chapter more. So understanding this thing is very important for the evolution genetics. Okay, now complete linkage will do what? Like for example, if what happened, me and him are together and we go to certain way or keep on going like this, we are losing lots of opportunities which could have made both of us more prosper. This thing is there. Okay, little bit we have tried to understand. Okay, it now how we will see the complete linkage? We will see that in case of certain features. What are the features? The features which we're gonna take it that there is the wings and the color of eye. Okay, so that is associated with one body type in the fruit fly. So we're gonna see the experiments done by the Morgans now. Okay, let's just see it into. Okay, so it can be observed. It can be observed in case of Drosophila. Since it's the scientific name, I have to underline it. Don't forget for your board examinations. I have seen that a lot of students don't underline it, line it. It doesn't mean that first chapter gone, your basic science also gone. No, everything will come back again and has to be applied. Okay, Drosophila, where gene of red eye and normal wings this is what wild type. Wild type word in itself means, for here understood that it is a dominant feature. Dominant feature means what? Even one allele of this will come, it will show this effect. Okay, so wild color, homologous. Homologous means that it has what? If eye color is this, we will be writing it capital cap. Okay, okay, this like this. Okay, and we will write this as any way you can write it, like capital and this is what? Small, like normal being capital. So this is the wild type. Okay, so I'm not writing it up as of now. Okay, we are just writing it up. Okay, so wild is, you won't be able to remember it. We will see it like capital B. Okay, so this homozygous condition, so homozygous female, okay, is crossed with homozygous recessive, because dominant character has already been taken, homozygous recessive. So recessive color is what? Purple eye and vestigial wings. Vestigial means which is not able to move. Okay. Vestigial wing male. Okay. So purple eye would be what? Small. Small. Vestigial is small v, small v. Okay. This is being done. So what we will see? So we see that the F1 generation would be what? The red eye and the, the first generation would be what? This is the dominant means red eye and the normal winged F1 generation. Now what we did? We took the one of this F1 generation and did what? Selfing and the test cross. And let's see that whether the things come up or not. Means what thing comes up or not. This thing comes or not. So we will be doing the one test this, one test this. Okay, so selfing and all those things will be done. Okay, so after the cross, okay, let's see that. So there is what? Parent. So I'm gonna make the cross now. Okay, so this is parent. Parent was what? The wild parent is what? We are seeing, making it like this. So these are genes present. So capital P, capital P, capital V, capital V. So this is what? This is a female, a wild type one. Okay, and the other one is what? Male, which is what? Recessive. So small p, small p, small like this. Okay, small v, small v. When they will make the gametes, finally, of course, everyone would be what? Same gametes will be formed. So the progeny, the crossing, I'm not making the gametes this time. So progeny would be what? One capital, okay, on both, and other would be what? Small. This leads to what? This F1 generation. The F1 generation will show you what? It will show all of them are what? They are red eye and normal wings. Okay, now what I did? Now I did the selfing and the test cross. Okay, so two different things I have done it. Okay, so first thing is what? Here I am doing selfing of it, and when I do the selfing, this is what? F2 generation. So in selfing I have what option? I had the dominant option, so I am showing it with the red color. Here also I will make it with the red so that you can remember. Okay, and this will also be a red. Okay, so this thing is there, a red color, and this would be okay, selfing we will be doing it. Okay, so that red color thing means capitals ones are coming, here the small ones coming. So when they will make the gametes, so what options they have? They have either this can form, this can form, this can form, and this can form. Let's see that what they are making up. Red, 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 and this is also red. This will be white, white, and white, white. So what happened if you see in a selfing? Okay, what we have said, come back, see it here. We said that if we would be doing what? If we would be doing the selfing. So this is what dihybrid cross shows. F2 generation means when selfing would be done. 3:1. 3 would be the dominant character showing, 1 would be the recessive character showing, right? So see here what happened. These 3 all are what? Capitals. So these 3 will show what? Dominant character, and this one would be what? The recessive character. So it is actually what? All are parental combinations. All are parental combinations. And what happened when there will be test cross? So in test cross, beta, what happened? In test cross, we take this parent. Okay, this is the F1 generation parent. This is F1 generation parent, and we take a recessive trait. So recessive trait is this, means this one only we are taking. Now what options we are having? So we have with the cross these options available. So that is what? That is 50% this, 50% this. Means still it remains what? The dominant and the recessive trait only, means similar like this only. Okay, means parental trait like this only. Okay, there is no new thing has been formed. Same thing, means that 50-50 means 1-1 ratio. Can you see over here? What is this? See over here, test cross. 50%, 50% means 1:1. Okay, so no new recombinants has been formed in both the cases. All are the parental combinations. So this complete linkage is seen in case of male Drosophila. Okay, when this happens. Okay, so this is what? Complete linkage. Now let's go to the incomplete linkage. Incomplete linkage. In incomplete linkage means little bit crossing over occurs. Okay, what it is we will learn it, and this is very common in nature. So first thing is very common in nature. Okay, F2 progeny. Okay, so the F2 progeny will show all the traits. Recombinants also will be there, but in a different proportions. Okay, F2 progeny shows both crossovers as well as non-crossovers in variable number, in variable proportions. Okay, number of recombinant is usually less than the dihybrid cross. Okay, so number of recombinant, because still there is linkage now, so recombinant are not able to come that much. Okay, so number of recombinant is usually less than expected in independent assortment, which is 10:6. That 6 is not there, less than that is there. Okay, so 10:2, something like this. No, less than that. Okay, so expected in independent assortment, and we see this in the female Drosophila. Okay, so the example is this one. Example was what? Seen in male Drosophila. And when it is doing, when the F1 males are test crossed. Okay, here what happened? In this case, incomplete dominance is seen in case of female. Incomplete linkage. Linkage is seen in female Drosophila. Okay, so we have what? We have red-eyed normal wing, which is, this is what? Wild type. This is what? Dominant homozygous female, same which we have taken earlier. Okay, we can even write it like PV also. Okay, so it is being crossed with purple eye, vestigial winged recessive homozygous male, that is small p, small p, small v, small v. Okay, so F1 progeny would be what? Dominant. Okay, so F1 progeny would be all heterozygous This thing is same only which we are learning in Mendel. Nothing new I am doing, so that's why I'm not elaborating it. Okay, so heterozygous, red-eyed and normal winged. That is what capital P, small p, capital V, small v. This thing is there, 100% will be them. Then what happened when this F1 generation test crossed with what? With homozygous recessive male. Okay, then it does not yield 1:1:1:1 thing which is seen. Okay, it goes to 9:1:1:8. Okay, it does not yield 1:1:1:1, means 25 factor is not there. Instead 9:1:1:8. These two are what? These are the parental combination. Parental. But these two which you are seeing, these are the recombinant. Means recombinant forms, but very less compared to this. But forms means it is incomplete linkage. Little bit chances are there. So what happened? This also shows that the genes segregate independent of each other. Here, means with the blue recombinant only I am writing. Here, gene segregates independent of each other. Independent of each other. And forms 90.7% parental combination along with 9.3% recombinant. Okay, means less, but it formed. Okay, so there might be different types of gametes that might have formed. So what happened in there? The same thing in the Mendel. So what happened? See, this is the female and this is the initially male. Okay, so what happened finally over here? They form F1 generation. The F1 generation would be like this. Okay, now this F1 generation, when it goes for test cross, so this is F1 generation. Now this goes for test cross. Okay, so test cross. In test cross, beta, what happened? This thing will be making a cross with the recessive one. Now in this case, what happened when it is in the male? In male, okay, it makes changes. Okay, so what happened? 4 different types of gametes form. Normally there will be only 2 types of gametes form, but here 4 different types of gamete forms. Question is why? Because in this case, what happened? Crossing over occur. Crossing over occur during meiosis. Meiosis or gamete formation. During gamete formation or meiosis, whatever you want to call it. So which leads to what? It leads to newer combination forms. For example, this is capital this was what? This is small p, small v. So the option forms is what? There will be capital P, small p, small p, capital V, small v, small v. So these are what? These are the gamete forms. Although these were lesser in number. That's why only 9.3% recombinant. I hope this thing is understood over here. It formed but less. This is incomplete dominance. Oh, sorry, incomplete linkage. Proper linkage is not there. New recombinant came up. Why came up? We have wrote it up. Next comes the important topic. With this incomplete linkage, complete linkage is over. Next comes the factors, which is the very important one. Factors affecting linkage. Now, factors affecting linkage, we have to remember what? First of all, age. As age increases, linkage also increases. And because of that reason, lots of disease also coming up. So, age is what? Directly proportional to linkage. That is, increase in age means increase in linkage, and less recombination. After age, the next thing is temperature. Now with temperature, it is reciprocal, inversely related. Means as temperature increases, linkage decreases. Means genes will separate apart, because just think that if me and my friend is there, and if the temperature, like in winters we will sit closer so that we can have little bit of heat, but in summers we will like, "Aya, get away from me, macha," like that. Okay, so temperature is inversely proportional to linkage, and means here this case would be directly proportional to the recombinant. More recombinant will be formed. Okay, because when me and my friend will be going away, new people will try to make the friends out of it. So that thing is there. Next, after temperature, next thing is radiations. Radiations also do the same thing, because radiations are bad, na? So imagine that radiation is the vamp of the cereal. So ting ding 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 ding. Okay, so if the radiation will come, linkage will be less. Okay, so means closeness will be gone. Okay, so linkage is inversely proportional to it and directly proportional to recombinant. When I'm saying to the recombination, it also means that directly proportional to crossing over also. So here also crossing over. Okay, then fourth would be what? Here it will be inversely proportional to crossing over. Okay, and the fourth is the very important, distance. More the distance, less would be the linkage. More closer, most close, links would be there. So it would be inversely proportional. So increase in distance between two genes means it will decrease the linkage. Okay, with this you have to understand certain other facts also. So next point is, what is the significance of this thing? Significance of linkage. So significance of linkage is that helps to keep the parental, racial and important traits together, because they are not being diluted into the next. Because in hybridization what happens, certain features goes to the next and it passes on. So some has that feature, some don't have that feature. Maybe I want it but I'm not getting it. Okay, like that. The next thing is maintains maintained important traits of a newly developed variety. So if the genes which I want, I will put it closer so that crossing over don't occur in them. Okay, then third is linkage groups gives information about the location of genes in chromosomes. and that we will do it with the help of a weight. Now the question is that, okay, linkage is there, linkage was always there. How Mendel never found out this? We said now unlinked genes will always go forward what? The Mendel inheritance. Why the characters he took never had a linkage problem? This is a good story. Okay, so let's just see that. Okay. So the question is that could be asked to you as an assertion reason based question, or it can even be asked in your board examination academically. Why Mendel missed linkage? And let's see it. Basically, so where the genes are present, let's see. So there is what, 7 chromosomes pair present, and the characters he took, 7 characters, they are present on 4 chromosomes only. Means there might be the case where certain genes which are present on the same chromosomes, because if only 4 chromosome has 7 characters, maybe some chromosome would be there where the 2 genes or 3 genes would be present, and there would be the linkage case, because that would be one linkage group till now, if you've understood. That would have happened. Let's just see that. So basically I'm making what? First I'm making the chromosomes. Okay. One. So this is what? This is chromosome number 1. Okay, completely I will write. Chromosome number 1. This is chromosome number 4. This is chromosome number 5. This is chromosome number 7. Okay, so these are the chromosomes on which the 7 characters were present. What were there? Now you have to remember certain thing, that on chromosome number 1, 2 genes were present. So basically 2 genes for the characters would be present. Then 3 genes were present here, means more genes were present closer, means more chances of linkage will be there. This is more linked groups. Okay, so then there is what? 1 gene is present on it and 1 gene is present on it. Okay, so if he would have taken a character from this and this, no linkage would be there. If taken one here, no linkage would be there. But if he would have taken the character from here, means 2 characters present on 4, then he might have encountered the linkage. But to the surprise, he never made that cross. So what are the characters present on this, which he didn't did it, was like this. So there was gene for seed color, flower color, these 2 which were so close present that if he would have done it, then there might be the case that he will end up in the linkage kind of situation and all his work would be diluted then before he even submitted up. So he never did it, or maybe he did it but he never showed it. He's clever. Okay, he's a monk, but he might be very clever. Okay, so these genes are closely linked, but Mendel never did cross on this characters, on this genes. Understood? So this thing is there. Next thing, so whatever he did it, crossing over occurs. We understood that. Now one question I am giving up to you before going up to, and we will try to discuss it. Okay, so what is the question that you have to tell me how linkage do not support independent assortment? Okay, so question is linkage versus independent assortment. Independent assortment. Explain, or means how linkage, same question if it could come, how linkage do not follow law of, or it's scattered law of independent assortment. Or in the better words, opposes law of independent assortment. This is the question for you, which you have to do it, and I will let you know if you understand meiosis from your 11th class. Definitely, if not, then wait for my classes, okay, because I will help you out with those. Next comes like, okay, we understood the linkage. Let's just see a little bit more about linkage, that is very specifically sex linkage. Okay, so we have reached to the topic called as sex linkage. Sex linkage means that we know that sex chromosomes are there. On the sex chromosome, certain genes would be present. How those genes are present and how closely those genes are present and affect the characteristics. Means, for example, I have Y chromosomes. Y chromosomes has 231 genes, right? Now, those 231 genes will be closely related to me from my dad. So the inheritance of my dad, which is present on 231, would be similar on me, unless there will be certain crossing over that would be occurring. Okay, so this thing we understood little bit. Okay, so me and my dad would be similar in those 231 genes characters. Okay, so the sex chromosomes X and Y carries genes for sex determination. And some other genes also, which is for the somatic character or vegetative character, means maybe like the eye color or something like that. Okay, and some other somatic, which is also called as autosomic, and sex character is known as allosomic. Okay, so autosomic characters. Now, genes on somatic characters which are present on the sex chromosomes are called as sex-linked genes. Okay, so genes for somatic characters present on sex chromosomes are known as sex-linked genes. Now, the characters which are controlled by it, okay, by the sex-linked genes are termed sex-linked characters. Okay, very simple terminologies. Okay, so characters which are controlled by sex-linked genes are termed as sex-linked characters. Simple thing. Okay, nothing math, basic terminologies. Okay, then inheritance of these characters, they are called as sex-linked inheritance. We are just using the words, nothing much. And there are diseases which we have to study. Two most important diseases that we have to study is that color blindness and hemophilia. Now, this sex linkage can be depending upon the different sex chromosomes. So, for example, it could be X-linked or it could be Y-linked. Okay, so there are two types of on the basis of sex chromosomes. Now we're going to be studying about X and Y separately and the factors and which are being done by X, which are done by Y. Okay, so let's just see. So first is X-linked. X-linked. Okay. Now in this, what we have to study? So certain things. So first example we are taking from the Drosophila itself. So in X-linked, first thing you have to remember that It is seen in case of white-eyed male Drosophila. Okay, so it is seen, later we will see it in humans also, don't worry. Okay, when we are doing the questions. Seen in white-eyed male Drosophila. Drosophila. Now let's take the example. So this is your parents. Okay, now for the male I have did what? I have made this sign. Okay, because here this eye color is a feature of sex chromosome. Okay, so it is present on the sex chromosome. So this is what XX of the, like I am taking it like a humans. Okay, XX XY. Okay, and this is XY. So this XY will make what? Male. Okay, so here what happened? This is the male and this is what? 2X means female. Now they have the white one. So means, remember that this X thing must be having what? It having a gene for it. Y do not have. So here I am writing what? X chromosome has gene for eye color. Now the dominant character is red. So means if the red gene will come single also, it will show effects. If double, definitely will show effect. Present on X. And the recessive character is seen in case of white only. Now for white gene, if it has to be come in double, then only it will be shown by what? A female, or else it won't be shown. Okay, now what happened? If initially the female is XX, this is what? Red. The red color chromosomes means they have a red gene present. So how I will show it? W plus W plus. This is wild type. Okay, red is what? Wild type. So wild will be showing with the positive sign. Okay, so it will be showing by this sign and this is by this sign. Okay, so the signs have been assigned. Okay, now here it will be what? It will be only W gene, because on Y there is no such eye color gene present because it's smaller, na? So small thing won't have anything. So this is what? Only W. So when only W present, it will show it effects. So this is what? White color eye is shown. This is red color eye is shown. Now what happened? They will make gametes. Okay, so this is what? Two different types of two gametes, and here also this sex chromosomes will divide. Now what will happen? There will be crossing over. Okay, so crossing over. So crossing over option is what? That anyone can go with anyone. So one option is this. Okay, another option would be what? To go with this only two options can be formed in their case. Okay, no matter, there will be multiple offspring, but two options is there. So this is what? This is male offspring. This is F1 generation. You can make it all the four. Okay, and this is what? Female offspring. This female offspring is what? It has this and this, means it is what? It has a white gene also present, which is coming from the male counterpart father, and it has capital red also, but the look would be what? It will be red-eyed. Okay, this is red eye, this is white eye. Okay, here the male would be what? Since the male white Y gene do not have any gene present for the eye color, this is only the X1. The X1 is red, so this male will also be red-eyed. So F1 generation would be what? When it is a heterozygous condition, it will be showing what? It will be showing a red color only. Okay, so this thing is there. Now they will make gametes. Since they are heterozygous, there will be two different versions will be formed. Okay, for this one. Now what will happen? This is F2 generation. Now F2 generation crossing over would be occurring. This has option to go with this. This has option to go with this. This has option to go with this. And this has option to go with this. So these are the four options. What 4 options? This is red-eyed female, because this is what XX chromosome. This is XY chromosome, XX chromosome, XY chromosome. Okay, so this is red-eyed female. This is red-eyed male because a male is there. This is red-eyed female, and this is white-eye male. What happened here is, are you seeing there is no female with the white eye? It is always the red eye. Now, if they two go into the breeding, na beta, here you understand, these two go, then there will be the chances that a white color female will come. But so less chances, mostly chances are others ones, right? So this thing, like, I'm not saying that this can't happen further and the female can't come up. But this thing, white color, is very much linked with the male because it can show only when it's in the recessive conditions. And that's why this becomes a very important conclusion for us. So 2 conclusion we got it, that the male is only coming up because it is a recessive trait which is present on the X only, but it can only show in this case when the male would be there. If male wouldn't be there, then there might not be any female also with the similar features. Okay, so this thing is there. So here what happened? Earlier there was a male. Wait. Earlier there was a male, then no one in this generation. Then again it comes up. So what happened? This the feature which is present here is actually reaches to another, which becomes carrier, and then it comes back. Okay, so means what? Male, female, where the gene is traveling. Gene is from male to female and then again to male. And again it can go to the female if it do with this one. So what is happening? It is a crisscross thing is being made. Okay, things are changed. So this kind of inheritance is also called as crisscross inheritance. Okay, I hope this thing is being understood to you. So this is what it is, a crisscross inheritance. That is, here what happened in the crisscross inheritance? There is a male, then there will be a female carrier, and then again male. Okay, so affected ones. Here female is very less chances of affected, because if the female has to be affected, it has to show this thing. Okay, so this thing is there. It can go vice versa also. Okay, so what are the characteristics of the sex linkage? Characteristics of sex linkage. Number 1 shows crisscross inheritance. What is crisscross inheritance? Means father to the daughter, then to the grandson. Okay, or maybe it could comes like what? Mother to the son, and then to the granddaughter. Now here, since this person is the carrier, so on that we will say that what is the terminologies for it. So in this case, since we are taking this, so for the daughter is what? Female. For female we use the word gynic. So here what happened? Diagynic crisscross inheritance. Whereas here the son is there. For the male we use the word androecium, andro. So this is what? Diandric crisscross inheritance. Because the male is the carrier, the ones which is carrier, which is changing, which is making the cross. Okay, so that is what? The ones which we are taking into the account. So remember this fact. Okay, then more to this, what is there? Normally what happened in this sex linkage? mother, like if we say that the first one, okay, the one that the digenic conditions which we have just studied about the Drosophila, okay, mother passes alleles to both son and daughter. See over here, this is what, this is the mother, okay, passing the alleles to both this also, this also. So it is passing the correct alleles only the father is giving something different, same alleles. So that thing, later on when this act as mother, it gives what to both, it is giving this color to the male also, it is giving this thing to the female also, it's not just like the one only, okay. So both mother and daughter, okay. Then the next thing is majority of sex linked are recessive characters. Majority of them, very less are them, but majority of our recessive characters, we have to remember. Means recessive characters means that if it has to show, it has to come in the homozygous conditions, or it has to come with the Y genes which do not have the counterpart present. So only the one which is left, it will show it effect. Okay, first thing here I want to tell you also one thing, that how the chromosomes actually look like. Okay, if you see a 6 chromosome na beta, this is what, imagine that this is one chromosome and this is what X, okay, and its counterpart, okay, see this is the counterpart, this is the Y. Now see the size difference, X and Y, just not the size difference. If you see that, for example, if the color, if the character is over here, its counterpart is missing because Y chromosomes do not have its effect. So what happened? The counterpart is missing, counter gene allele missing. So what happened? It leads to what? Leads to single allele to express. Now here comes a very important question. What it is? That sir, we always says that when we were studying the chromosomal theory, that alleles always occurs in pairs and it expresses in the pair. How come this is expressing? And this is your question of the day. Okay, so what is the question? The question is that how single allele expresses here. Okay, this thing is there. I hope this thing is a little bit of conceptualized and you are able to understand better. Then the fourth point is, this is more apparent in males because its counterpart is not present in them. Okay, so more apparent in males. And I told you what females would be, what? Females would be carrier. Females are carrier. So what we say, that sex linked effect, male will be more affected, and recessive gene, if there is, the male will be more affected. If the dominant gene is there, then female would be the affector. So sex linked recessive gene is there. If sex linked recessive gene is there, then male will be affected because male do not have the counterpart of it, only one X is there. Okay, if sex linked dominant gene is there, means it is in the dominant state, then the female will be affected because dominant gene will show effects in single also, double also, but for XX it needs to be in the dominant cases. Okay, so female will only be affected if it will be in the dominant cases. Okay, next with this, your X linked is over, Y linkage is there, which is very small and easy. Y linked. So usually Y chromosomes do not have much genes and much effects. Okay, so you have to remember that there is a one thing, that sex determining reason What is sex determining reason? It is a gene which actually expresses itself during the initial weeks of the gestation, and that start releasing what testosterone, and then the male character forms. If that gene is disappeared and not present, then the female fetus will form. Okay, sexual reproduction in human beings, we will learn it over there. So usually what happens, that happens. Okay, so that is usually Y chromosome is considered as empty chromosome, as it contains very less number of genes, that is 231 in humans. In humans, of course, in humans. Now, and important one is what? Testes determining factors, or which lies on the sex determining reason. Okay, so TDF, that is testes determining factor present on SRY, that is sex, okay, SRF, sex determining reason of Y chromosome. Okay, so these genes which are directly inherited from the father to me, that is what I was telling you. Those genes are called as holendric genes. Whole means whole, completely. Andric means father, okay, male. Characters are being taken up. Okay, so that, for example is that hairs on the pinna. You have seen that certain males has hair on the pinna. Their child will also grow that if they have it. Okay, so this thing is there. So these are called as holendric genes, where what happened? It goes from father to son, father genes to son. Okay, example, hair on pinna, and this is called as hypertrophy. Okay, or baldness is also the character of dad. My dad is not bald. I might not got bald unless I have put lot of gels and waxes on my head. Okay, hair on pinna condition is called as hypertrophy, or maybe the baldness. So male pattern baldness is being seen. So you might have seen that certain uncles and their son has this problem. In your family, if it is there, do let me know in the comment section box. Same holendric genes now. There is hologynic genes which goes from mother to the daughter. The term you have to remember. Okay, that's, it's not in much in syllabus. Okay, so hologynic genes are what? Which goes from mother to daughter. That's it. Next thing is certain abbreviated things which we have to remember, that is like sex limited characters. These are what? They are certain characters which are limited to one sex only. Okay, for example, like memory glands development in female. Male do not have it. The male still have in the rudimentary forms. Okay, so that is what? Milk secretion in female. Okay, then there is what? Pattern baldness in the male. So male got this but female don't got. Okay, so pattern baldness in male. Next thing that we will do it is sex influenced characters. Means that certain characters which are more pronounced or more influenced in a particular sex, gender. Okay, so they are controlled by the sex hormones. These are not characters. Sex limited characters are genetic characters. Okay, so sex influenced characters. So they are what? Certain characters which are controlled by sex hormones. They are what? Controlled by sex hormones and express better in one gender. Example, low pitch voice, beard, mustaches in the male. Example, low pitch voice, beard, mustaches in males. Now you will say that, sir, I have seen that this girl sitting next to me has certain mustaches over here, certain beard also there. So what is this? So better remember that in our body there is both the hormones present. Means in my body testosterone is also present, estrogen is also secreted. Then you're like, you sir, you have ovaries? Like no beta, I don't have ovaries present, nor do you. In your body also it's occurring. The reason why they are present because of your adrenal gland. That is from 11th class topic, that is chemical coordination chapter. So in that what happened, your adrenal gland, which is present over your kidney, it secretes both estrogen and progesterone very less amount. Okay, and when it is secreting that, it's giving the effects. So sometimes what happen in the body, if in the female estrogen level is less, what happen, that leads to the release of like marked effect of testosterone. Okay, and what happened because of that, certain these simple things comes up. Okay, this is nothing to be worried about, females, you are all okay. Next thing that we're gonna be studying is about gene mapping or chromosomal mapping. Okay, so the next is gene mapping. Okay, important of the all. Okay, this is yeah. Gene mapping, it is also called as chromosomal mapping. Means whole of the chromosome we are seeing that which gene is present next. Okay, chromosomal mapping. Okay, so it is what? It is the method of asserting the position of the genes on the chromosomes. Okay, so it is what? Asserting the position of genes on a chromosome, known as what? Known as gene mapping or chromosomal mapping. And it depends upon the frequency of crossing over. Means frequency means chances of crossing over. If the chances are more, then they are far off. If the chances is less, then there is what? It is less. Means it also depends upon the distance. So crossing over is more, means distance is far off. If the crossing over is less, okay, means distance is less. Okay, so this thing is coming. So what happened? Chromosome map is prepared on the basis of frequency percentage of crossing over between two genes. Means distance is very important, frequency is important of crossing over, linkage is also being estimated with it. Okay, so we have learned the factors, so that factors thing will be coming up. Okay, so what is that? We know that distance is what? More the distance, less linkage. So inversely proportional to linkage, and more the distance, more the crossing over. So means directly proportional to the frequency of crossing over. Okay, so we can get this fact understood. Okay, let's just see that. For example, we understand it. For example, if frequency of crossing over between gene A and B is 20%, then that means that these two genes have a difference of what? 20 centimorgans. Means it is being, how they are, like this thing is being measured. It is measured in certain units. The unit is centimorgan units. Okay, so what happened? That means two genes are 20 centimorgan units apart from each other. And I remember it at 20 centimeters. So they are 20 centimeters away from each other. Okay, this far. 20% chances hai, okay, to cross over. Okay, so 20% they are away from it. Okay, let's just do a question on it, and that becomes a very important factors. So what is the question? I am giving you the questions. If the crossing over frequency of gene A and B is 28%, B and C is 20%, and A and C is 8%. Okay, map, or what will be the sequence of these genes on chromosome? So now you have to tell that which gene will come first, which will come second, which will come third. So here your critical thinking will work. Moreover, your reasoning based questions which you might have if appeared for Olympiads, very good. If not, then we will do it here. One question I am helping you here. Later questions when we will do in the practice sessions, there we will do it. Okay, so let's see that. So it is saying that, like, we have to remember that we are doing what? Scale. So we are playing with the scale. So let's take it the scale only. For you right now I am giving you the scale. Okay, in reality nothing like that. Okay, so this scale, okay, not very good. Okay, think like that. So it says that A and B is 28. So like this, what? So if we say that A, if this is the A, then where will be the B? This is B. This distance would be what? 28 centimorgan units, because percentage means 28. So I wrote it 28. Now they're saying that B and C is 20 centimeters. Now C, now what option is there? I'm changing the color. Two option is there. Now this can go like this. One option, first option is this, that this C goes here, it becomes 20 centimorgan unit. Or it can go what? Second option is that it can come over here, somewhere here. So this is also what we are imagining, that this is 20 centimorgan unit. This is second. Options are there. Two options are there. Let's just take both the option and see that which one will be correct. Okay, so I'm making it like this to make it easier. So from first statement we have made it this. Now the second statement. Second statement is this. Okay, from this statement we did what? Already formed thing. We will be writing it like that way. So till now we know that A, B is there. Now option is one, first option. First option is it can go like this. So C comes here, 20 centimorgan units. Okay, or second option is there what? That A, B and C comes something here, because it is not showing that it is minus going back or there. Just the percentage difference between two genes are being given. So C can be here. So this is also 20 centimorgan units. Okay. Now let's see the third statement. Third statement says that A and C should have 8 centimeter. So third statement is A and C should have 8 centimorgan units. If you see over here, the first option, here A and C has what? This distance plus this distance means 28 plus 20, which becomes what? 48 centimorgan units. So here this case, in the first case, A and C becomes what? 20 plus 28, that is 48 centimorgan unit, which is not the case because we need 8 centimorgan unit. But if we see here, so this is 20, if you see this area, so this is what? This whole thing was 28. So this whole thing was what? 28 centimorgan units. So this is what? This big one, small one minus, that is 28 minus 20, that is what? 8 centimorgan units. Means this is what? 8 centimorgan unit, which is similar. So means this becomes the sign, okay, or this becomes the correct answer. So that is what? A, C, B. So gene would be present like this way. First A will come, then C will come, then B will come. So this becomes a correct answer. I hope this thing is understood to you. If you understood to you, do let me know in the comment section about it. Well, that's all for this video of linkage and recombination, and we have completed pretty much everything about it. And we will be like now you go open your NCERT and see the page where they have given you like in complete linkage, two different options, two different way of seeing it, okay, two different way of basic to the advanced version of the linkage and the disputes in the linkage. Okay, now if that thing is completed and you have understood it, well done, clap for yourself, you have understood the linkage and the recombination topic. This topic has certain link with your 11th class crossing over topics from the cell division chapter. Well, if you like this video, give it a like, share it with your friends. Don't forget to subscribe because it motivates me a lot. Well, that's all for this video. Thank you.